moving on to mitosis, meiosis, and gametogenesis. So we all know about mitosis, right? And we'll talk a little bit, real briefly about this. We had a really good run of that in general NP. There's no need to really reinforce it a ton here. I will briefly mention it. There are some, there is a microscope slide in the back that's dialed up with these different phases that you're to know. So just think back when you did general NP, and we'll kind of look at that briefly again. Meiosis will be new for you. This is sex cell division. So we're talking about the sperm and the egg and dividing the chromosomes and making them genetically unique. And gametogenesis is the formation of these sex cells, if you will. Okay. So being the sperm and the egg. This is kind of what we're covering here today. Where are you all at in lecture? Did you guys start any of this stuff yet? Okay, so you just kind of started to get into that, I would imagine, probably not in too much detail, right? So you guys have already completed your DNA protein synthesis, because you guys have a test every week, is that right, lecture?
Yeah.
Okay, so you're moving pretty quick, right? Okay. All right, so we'll start with objective 1 here, and we're looking at what is the difference between a sex cell and a somatic cell. So a somatic cell is what we call our body cells, if you will. So these are all the cells in your body with the exception of your sex cells. So in males, that are sex cells would be the sperm, and females it would be the egg, okay? Every other cell in the body outside of those two cells are what we call our somatic cells. Somatic cells are also known as diploid cells. Sometimes abbreviated with a 2N, and they have 46 chromosomes within their cell or their nucleus. Okay. And a sex cell is also known as a haploid cell, abbreviated with a small italics N, and they have 23 chromosomes. So something we'll get to after next week, after genetics, we're going to get into reproduction, reproductive physiology and things like that. And one of the things we get into is this cell right here called the zygote. So when you have the mother has her egg with 23 chromosomes, the male has his egg or his sperm with the 23. When that fertilizes, you get your very first body cell, 46 chromosomes. That's your very first somatic cell, right? And that very first somatic cell is known as a zygote. Okay? So this is the very first cell after fertilization. This is your very first somatic cell. But we'll be touching more on that in a couple of weeks from now. So being that this is our very first body cell, these cells here go through the process of mitosis. Okay? Any somatic cell that has DNA goes through the process of mitosis. Whereas the egg and the sperm, they're going through the process of meiosis, which is sex cell division. So a little different there, as you can see. And that's kind of what we want to get into, is differentiating the difference between mitosis, meiosis, and whatnot. So this week we do have a pretty good amount of microscope work that is dialed up. I did put some photomicrographs in the PowerPoint for you that you'll be able to see over there with some of the microscopes that we have on file. Okay. So this is where I didn't want to really go into a lot of depth in mitosis because we could spend a good amount of time on here, but being that the general view is a prerequisite, I do have some expectations that you understand mitosis, if you will. Remember, mitosis, that word really means the division of a nucleus. Okay, the division of the nucleus, but the division of a nucleus in a somatic cell, a cell with 46 chromosomes. And I don't want to really belabor this too much, but remember we have interphase, and I'll just briefly describe what this is. Interphase is the rest period of the cell. What this means is that during interphase, the cell is not going through mitosis. Okay? So 90% of the time a cell is spent in this phase, interphase, where it's just kind of resting, it's kind of doing its everyday kind of work, and then at some point it has to go through division. Okay? whether a cell next to it dies, whether your skin cells slough off, it has to go through this division. And then it goes into mitosis, which is division of the nucleus. And then it has those phases that we're familiar with, prophase, anaphase, metaphase, telophase. And then after it goes through that process, it then finally splits the cytoplasm, which is called cytokinesis. Cytokinesis is division of the cytoplasm. And what do I mean by that, division of the cytoplasm? What I mean by that is when this cytokinesis takes place, the cell actually physically separates into two new daughter cells. Okay, two new daughter cells, that's what we call them. But this is the key with this. When you get two new daughter cells, they have the exact same DNA. And why am I stressing that? Because in meiosis, we have division of the sex cells, but their DNA is not the same. It's genetically unique. It's different from all of them when they go through this process. So we have to compare and contrast the differences between mitosis and meiosis. So on a microscope, you need to know the phases of mitosis and interphase. And we do have one of our scopes back there dialed up. I don't have anything on here because, again, I don't want to spend too much time on that. We have photomicrographs or models as well. We do have the models over here of mitosis, I believe, is what we have there. This might be meiosis, actually. I'll look to see if we have the mitosis slides. They're probably over here somewhere. Okay, but any questions on mitosis at all? Okay, so hopefully that's kind of old news for us. So we get into meiosis, and this is sex cell division. And so the end result with meiosis, if you think about mitosis, the end result was two genetically equal daughter cells with 46 chromosomes. And with meiosis, the end result is 4 haploid cells with 23 chromosomes. And, like I said earlier, genetically unique from one another. All 4 of them are going to be completely different from one another. So with meiosis, we have 2 phases or stages, if you will. We have meiosis 1 and we have meiosis 2, and we need to know what's happening in each of those. Okay? There are of course some parts of this meiosis 1 and meiosis 2 which are extremely important, and the other phases aren't as important for us right now, but we'll see that as we go along. Okay. So here's just kind of a real quick, like, dirty schematic of it, of what we're going to actually be looking at. So all our sperm and our eggs are going to come from a parent cell, which, as you can see, is going to be a diploid cell with 46 chromosomes. But there is each male and female are going to have the same process of meiosis 1 and meiosis 2, but there are going to be some differences between male and female. Okay? Males are fairly straightforward. Females, there's a little subtle difference with that. Okay? And we'll look at that as we go along. But the bottom line is that we start with a parent cell, and these are what we're going to call undifferentiated, write that down, undifferentiated germ cells. Okay, that's what we call them. Undifferentiated germ cells. That's another word for this parent cell that has 46 chromosomes. In the male, we call this undifferentiated germ cell a specific name, spermatogonia. So write that down. Spermatogonia is the specific name for this germ cell in the male. And in the female, it's oogonia. And we'll talk more about those as we get into each, the male and the female. But these are these undifferentiated parent cells or germ cells that have 46 chromosomes. So what we're going to see here in a moment is that this parent cell is going to reduce its chromosomes in half, 23, that's the little n, and then they're going to go through meiosis 2 and continue to divide even more, and we're going to end up with 4. Now there is going to be a difference between male and female with this. Here's going to be the difference. In a male, males are going to get 4 functional sperm cells. Okay? So what do I mean by functional sperm cells? What does that mean to you? What is functional?
They work.
They work, right? They're going to be able to fertilize, because that's their purpose, right? They're going to be able to fertilize the egg. So there are 4 functional sperm that will come out of this. Now with females, you are only going to get 1 functional egg, or ovum as we call it. Okay? The other 3 are going to be what we call polar bodies, which are non-functional. So there is a big difference between male and female. Males, we get 4 functional sperm cells. Females will only get 1 functional egg or ovum, as we call it. Okay, we don't use the word egg in here. Okay, I'm just lightly using that right now, but you won't see it in your lab packet. You won't see it on the test. It's not an answer you should put on the test. Okay, so ovum is an actual egg. Yeah.
Were the 3 that don't they call them here?
Polar bodies, yeah, and you'll see that come up. So we're going to get into this a little bit more, where you'll see those names and kind of how this all works, but they're called polar bodies. Okay. So we start with this meiosis 1. So the first thing that's going to happen is this parent cell here is going to duplicate its chromosomes, right? Kind of like mitosis did. If we're going to make new cells, we have to duplicate the chromosomes in order to give them to the next round of cells, if you will. So as you can see here with meiosis 1, the first thing that's going to happen is we get duplication of those chromosomes in that germ cell, okay, in that parent cell, if you will. And then we go through meiosis 1, which would be prophase 1, metaphase 1, anaphase 1, and telophase 1, okay? But prophase 1 is by far the most important part of them all for what we want to study, okay? Because look what's going to be happening here. The idea here is twofold. We want to reduce the chromosomes, but we also want to make them genetically different, okay? This is why your brothers and sisters all look different than you, unless you have an identical twin, okay? So what's going to happen here is the homologous chromosomes. What does that mean, homologous? Anybody know? Did you use that language at all in lecture at all? Homologous means the same chromosomes, okay? So when you think about a parent cell that has 46 chromosomes, 23 of them are for mom, 23 of them are for dad, and they should pair up, right? Making 46. Okay, so when they duplicate, then those 2 sets, if you will, are homologous, and they're going to join together. And when they do that, that joining together is called a synapsis. So when they join together, these homologous or the same pairs, and I'll show you a picture of this in just a minute, it's called the synapsis. And when they form, we call it a tetrad formation. So let's take a look at what I mean by that. So here you can see there is 1 chromosome right here. Here's the other chromosome here. These are homologous pairs to one another. They belong together. And so what you can see is they came together, all of these did, okay? And they created, or they synapses, okay? They came together, that's a synapsis. And what they formed is this whole structure called a tetrad. Make sense? Okay, so the process of them coming together is synapses. The formation now is a tetrad. So that's what's happening here in prophase 1. Now, what else happens is we get this. Crossing over occurs. So what they're doing here, take a look, they're swapping their genetic material. So look at this one right here as an example. You see the blue up here, red here, but then when you come down here, look at this. You follow this blue, and all of a sudden it turns into red because this genetic material from this chromosome swapped over, and you can see this red one here turns into blue because this genetic material came over here, and that's crossing over. And that's making these chromosomes genetically different. Would you all agree? This is very unique. Now, in mitosis, do we see any of that stuff going on? Not one bit. Not one bit. Okay? In mitosis, we just simply duplicate them, then they come together, don't they line up on the equator, then they split apart, and then you get two new daughter cells, which are genetically the same, and now we have all this crossing over, so we have a lot of genetic variability. So, we haven't even gotten out of prophase 1, and we've had all of this stuff happening. Now, what about metaphase 1? The homologous pairs line up on the equator randomly. That's a key word, randomly. There is no say as to where they go. And we call this independent or random assortment. They just randomly line up on that equator. That is going to give us yet another form of genetic variability later down the line. So I think I have a picture of that right here. So here's prophase 1. They form the tetrad. They do the crossing over thing here. And then look at, they're lining up in the center like we would have seen for metaphase in mitosis, but very random and genetically different. So then what happens here is anaphase, just like you remember in mitosis, they split apart. Well, what do we see here that's different that we don't see in mitosis? What do you see there? So here is anaphase in mitosis. And here's the cell. We had metaphase where they would all line up, right? And then anaphase in mitosis, they would split apart. So what do you see here compared to there? What do you see happening?
Centromere is still intact.
Yeah, the centromere is still intact, the whole chromosome is still intact, right? They haven't split apart. The homologous pairs have separated, haven't they? But not the actual chromosome, right? Interesting. So now we have 23 chromosomes here and 23 right here. And that's the end of our very first meiosis 1. So if we go back to this slide that I had over here in the beginning, this is where we're at right here. Now because they didn't split apart, this is where it gets kind of a little confusing for some people. Because they haven't split apart, separated those chromosomes, we still consider this 23 and this 23 over here because they have not separated. So technically these are haploid cells. These are with 23 chromosomes, yeah.
So why did they have to switch, like, the crossover?
Why do they cross over?
Yeah.
Well, this is just unique to sex cell division because if you think about, well, if you're asking me why, I can't explain why our bodies do that. Do you know what I mean?
Well, yeah, but like, so
I-
when they split or whatever,
Mm-hmm.
like,
Right here, you mean?
yeah,
Yep.
do they still have that small section that's different?
Yes, they do. See right there? Okay.
Oh, okay.
Yeah. So then they're going to go over, okay? And now you've got some of them genetically different
Oh,
here,
okay.
some of them genetically different there. But this is the really important part. These two are both haploid cells now. Even though technically there is 46 chromosomes, because they're together, that would make them only one. So therefore, it would be 23 apiece. Make sense?
Yeah.
Okay, that gets a little confusing there. So these are two haploid cells that we're making at the end of meiosis 1, genetically different than one another. Okay? Now, what we want to do is we want to reduce these even further to make 4 true 23 chromosome cells. Does that make sense? That's our next phase here. Okay. So this is where we see meiosis 2. And really there's not a whole lot, it looks and feels a lot like mitosis at this point. It really does, okay? So take a look. We have our, really says 23 chromosomes, but really we have 46, right? But we always say it's 23 because they haven't split. And they go through metaphase, look at, they line up on the equator. Then look at anaphase 2. Now doesn't that look a lot like mitosis now, right? Okay. So we've got 23 going to one side, 23 going to the other, same right here. And now these are 23, 23, 23, 23. These are your end result, 4 haploid cells with 23 chromosomes, all genetically different.
And because of the crossover out in the
Because of a few things, actually. Because of the crossover and the random assortment in metaphase 1, yes. So we've got two things that are causing this genetic variability. Questions? Anything I can go back to? This gets a little confusing when you got all these chromosomes running all over.
I have a question.
Sure.
It doesn't really matter, I guess, but, so you go from 23 weeks, meaning why doesn't it go to like 11, 12, or something? Like how is it still 23, and which one of those 4 when it's 23? You know how you keep splitting it up?
Oh, you mean like when it's coming down this way and it splits up here? Well, it just is, I'm not sure if I understand the question correctly, but basically, you really have 46 chromosomes in here, and so 23 are going to go to this cell, 23 here, same with this one, 23, 23. I guess I don't know that.
It was kind of a bonus question. But
Sure, that's okay. But yeah, so again, the whole goal of this though is to get 4 haploid cells with genetically different material. So just to recap that again, in that very first phase, prophase, you want to know where these are, or when these things are happening. That is some of the questions that we're going to ask you, of course, like when would you see synapse in the tetrad formation, prophase 1? When would you see the crossing over? When would you see this random assortment? When would we see chromosome reduction? We first see it, right, in telophase 1, and then we see it again in telophase 2, right? Okay? So we want to be able to know these really key characteristics of meiosis. When are they happening? Okay? So, just one of the objectives was to compare mitosis to meiosis. So these are in your lab packet. My page is off because they switched the lab packet since I last taught this, but you have a table in there, and then also you can find this in your book. But you can see mitosis up here. Look at, they both come from a parent cell. They both come from a cell that has 46 chromosomes, okay? But there's a difference between that because we call that, in the meiosis, we call that parent cell in the male spermatogonium or in the female ovogonium, okay? And so you can see this here with mitosis, kind of lining it up here. We get 2 daughter cells with same genetic material, no genetic variability at all, and there's 46 of each. And again, with the meiosis, we get genetic variability and 4 cells of 23 chromosomes. Okay, so we have some terms here that you need to know as well. So we have gametogenesis. Anybody know what that's referring to? This is sex cell formation. So the whole process of forming an egg or forming a sperm, or the sperm itself, is called gametogenesis. Again, this is sex cell formation. And the questions that we'll probably want to know is where do these take place? Where does gametogenesis take place? It takes place in the gonads, so that would be the testes and the ovaries, respectively. So spermatogenesis is the formation of the sperm. And spermatogenesis, the formation of the sperm, is taking place in the testes, but if we looked at it a little bit more in detail, inside the testes are the seminiferous tubules. Now, that is something we learned about back in general AP. I am going to show you a photomicrograph of that in just a moment. Oogenesis is the formation of the eggs or the ovum, okay? The ovum or the egg. And this is only taking place in the ovaries. Then we have a word that looks a lot like the first one, spermatogenesis. This is called spermiogenesis. And this is the maturation process of the sperm. It is the maturation process of the sperm. And this also takes place in the seminiferous tubules. So the sperm are produced in the testes of the seminiferous tubules. They further mature, called spermiogenesis. We'll look at that in just a moment here. So on Objective 8, it asks us to identify the following on a model or a microscope slide. And that's where you're going to get some time to do this. And once we're done, we have a small worksheet, not only one worksheet here today. And then we have the models that we can pass out and take a look at and get under the microscope and stuff like that. So what I've done here is I've put in parentheses behind each one of these a large M, which would indicate you can find it on the microscope, so you know which ones to look for under the microscope. And the other parentheses here, you see diagram or model, so you know not to look for those under a microscope, okay? That makes it a little easier. Because I don't think in your lab packet they differentiate which ones you can find on the microscope and which ones you shouldn't. So this will be a nice little cheat sheet for you. Okay. So these are the things that we're going to look at. I did put some photomicrographs up here that we'll look at here in just a moment. But we're going to start with the male. Okay. So we went through the whole basic concept of meiosis and what we would get at the end. But now let's apply that to both the male and the female. Okay, we'll start with the male. So up here now this is all taking place, yes, in the testes, more specifically the seminiferous tubules, okay? So if I drew you this seminiferous tubule out, if we were looking at it, so we all have an understanding here. If this was the tube, right at the very top, our spermatogonium cell, parent cell at the very top, that would be starting way out here on the periphery, okay? And then this is where the lumen, remember what a lumen is from the general AP, this is the space that things travel through, okay? And so eventually the sperm, as we can see here, are going to mature right into that lumen and then potentially be ejaculated out, okay? So if we look at this whole process here, all of this right here is meiosis happening, okay? So we come down here, we have our spermatogonium cell, which is the parent cell. It further divides, and we get a primary spermatocyte. This has 46 chromosomes, so these are things you're going to want to know, okay? Suppose the primary spermatocyte does have 46 chromosomes. Has it gone through meiosis yet? And how do you know that? Well, that's the end result of meiosis, but you got meiosis 1 and you got meiosis 2, because at the end of meiosis 1, you don't have 4. Only at the end of meiosis 2. So how do you know that this primary spermatocyte has not gone through meiosis yet?
So it's 46 chromosomes.
Very good, because it's got 46 chromosomes. Might seem elementary, but I just want to check your understanding of that, okay? So all of this right here, from this spermatogonium to the primary spermatocyte, that is just mitosis has occurred, right? That's it, okay? Now here's where we get to meiosis 1, right here. So we go from that primary spermatocyte, it divides into our secondary spermatocytes. Now, this has gone through meiosis 1. So think back to what we talked about. What happened in meiosis 1? We had the synapses, we had the tetrad formation, we had the crossing over, we had the random assortedness, right? Okay? So right here, don't we have a significant amount of genetic variability already? Okay? So these are our secondary spermatocytes with 23 chromosomes. Then it goes through meiosis 2, boom, 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 and there's our 4 that we were looking for at the very end. Okay? Look at here. This is spermatogenesis in the early phases. We have it in the late phases here. So what happens then is all of this, after the last meiosis 2 is complete, this is the spermiogenesis. This is the maturation of the sperm, isn't it? Okay? This is spermiogenesis. So these are called spermatids because they're not mature. Make sense? And then they mature. Now when we talk about them, we sometimes refer to them as spermatozoa. Same thing as sperm, but a little bit more of a formal name for them, spermatozoa. So if you see that on your worksheets or anything, you can relate to that. So this is the male. The male is really straightforward, right? Would you agree? I mean, it pretty much was just like meiosis 1, meiosis 2, and boom, we've got these sperm and we're good to go. Females, a little different, okay? And we'll get to there in just a moment. So here's a photomicrograph. This is something you guys saw back in general A&P. And this is a picture of the testes, okay? And these round little circular structures right here, these would be the seminiferous tubules, which we were drawing up just a moment ago here. See all this little swirly bob stuff in the middle here? That's the sperm. Okay? You're not going to be able to make out the sperm like you see it on a picture. We don't have the magnification for that in this classroom. So we're not going to be able to see a sperm under a microscope as in detail as we saw it before in the book. Okay, now what we're going to do right here in this next slide, we're going to go to the periphery right here of one of these seminiferous tubules. Okay, that's where we're going to go right now. Boom. Okay, so look what's on the periphery right here. Our very first parent cell, spermatogonia. And that's what you have to know. Notice it's hugging this wall of this seminiferous tubule. That's where we would find it. Okay. Let me go back to this picture. So this is the cell we're looking at. And then it goes through mitosis and develops our very first primary spermatocyte right here. Let's go back up to that slide then. Here it is. It divides. And a little further away from that wall are these larger cells right here, which are the primary spermatocytes. Still have 46 chromosomes. Okay. Anything I can go back to? Because we're going to switch gears to the female. And it's a little different. So I want to make sure we get your questions answered or you understand this before we move to the next little section. This whole thing right here, the whole process itself, generally speaking, for both male and female, is called gametogenesis, the formation of sex cells. This being just the males, that's spermatogenesis. And when we get into the female next, that's oogenesis, okay? So we've got to make sure we keep all those terms separate. All right, let's take a look at the female. So again, I put in parentheses here, microscope, microscope. A lot of this stuff, for the most part, with the exception of these two right here, you've seen on our general EP lab. Okay, so here's the difference here, okay? So this is our female. A little different. before birth, in the womb of the mother, the oogonium cells, the parent cell, have 46 chromosomes. And they turn into a primary oocyte before birth. Okay, so females, when you're born, you no longer have any oogonium cells. Males, we still have spermatogonium cells. Females do not. So that's a big difference between males and females, okay? So again, females, all of your oogonium cells turn into primary oocytes before you're even born. So what that means, females, is your eggs are all ready to go before you were even born. Where males, we're constantly making them. Does that make sense? Okay, from the spermatogonium cell. So in female ovaries, there is no such thing as an oogonium cell once you're born. The only thing that you have left is a primary oocyte. Okay? So then everything halts. You can see it says arrested in prophase 1, that means stoppage. So once you're born, you have all these primary oocytes, and everything just gets shut down until puberty. Okay? Once puberty hits, then we're going into meiosis 1. So here's our primary oocyte. It has 46 chromosomes. It goes through meiosis 1, tetrad formation, the synapses, the crossover, the random assortment like we talked about, and then it splits. So this is the end of meiosis 1. We get our first polar body, and we get a secondary oocyte. Now look at the difference there. Which one is going to be functional? Julie? Which one is going to be functional between these two? Right, the secondary. This one really has nothing, right? This is a non-functional polar body. It's not going to be fertilized. It's useless. Okay? But this one right here, the secondary oocyte, that does have potential. Okay? That could be fertilized.
Another question, so once puberty hits, then that sore is just thicker?
Correct. So when you go through puberty, when females are going through puberty and beyond, once a month, one of these primary oocytes is selected to go through this process. From the right ovary, and then the next month the left ovary, and then the right, and then the left, every month, right? Okay? One of them is selected to go through this process of meiosis 1. Now, it then goes out, the first polar body goes through meiosis 2 and makes 2 additional polar bodies, which again are non-functionable. So really, this whole side over here is really just wasted. Okay? We don't do anything with them. But they go through the process of meiosis 2. Okay, now here's the other little caveat here. Females, when you ovulate once per month, so that one is chosen, it goes through this process of meiosis 1, and then you ovulate it. Look at this, before you ovulate it, it arrests in metaphase 2. It stops. Okay? And only if the sperm fertilizes it will it complete meiosis 2. Only, that's really huge, that's a big point. Highlight that, circle it. Only if that egg or the ovum is fertilized will it complete meiosis 2. So look what happens. It gets fertilized, there's the sperm. There's the sperm, it fertilizes it. It then goes and completes meiosis 2, and it creates a secondary polar body, which is non-functional, and our ovum, which is functional. And here we are, 1, 2, 3 worthless polar bodies and 1 functionable ovum. Only after it's been fertilized. That's the key. So you can see there's some differences here with the female compared to the male in regards to that. Questions on this? This is not always easy to follow, so let me know what I can go back and refresh you on.
What is it get arrested on if it doesn't get fertilized? What is it?
So if it doesn't get fertilized, it will arrest in metaphase 2, and then females will just, during the menstruation process, it will just bleed out. It will just be wasted away, if you will. And that's it. So really the only time it matters that it goes through or at least meiosis 2 is if it's fertilized.
So that is our 1 functional ovum then?
Correct. Yep. Yes.
Do you just call it the oocyte until it gets fertilized, then it's called the ovum?
Correct. Yep. Yep. Once it gets fertilized, then we call it an ovum. And it's got 23 chromosomes from mom, and then there's 23 from the sperm of dad. And then those 2 are going to eventually combine, aren't they? And then what was that name of that very first cell that we talked about? The zygote cell, right? That's what this will become. The ovum will become the zygote cell, and the sperm and the egg unite the chromosomes. Okay. What other questions do you have? Look at this right here. There's a little bit more to the objectives here in regards to this, because over here you're going to notice that the egg here, or the egg or the ovum or the oocyte, will have a follicle around it, okay, as it's going through this process. And then when it ovulates, it only ovulates the oocyte out, and it leaves the follicle behind. And the follicle becomes this structure here, which we're going to talk about in just a moment, called the corpus luteum. Okay? So we'll look at that in just a moment. So if you look here, one of these primary oocytes right here are chosen to mature in the ovary. And at about what day does ovulation take place? If we were looking, remember back to our general EP days, I don't expect you to remember it, but about what day? Out of a 28-day cycle, 14, right? 14, about halfway, right? Now, is everybody in here going to be different? Yeah. You're not ovulating right on day 14. I always make that little disclaimer in my class. Don't plan your life around this, your family planning around this table here, okay? Don't come back to me saying, you told me I was going to ovulate on day 14, I never had a baby, or vice versa, okay? So, but this follicle here, this follicle is called a mature follicle. We're going to look at this in a moment. But that goes on to become this structure here called the corpus luteum. We're going to look at that too in just a moment. Okay. So what do we have here? This is something you have to identify. This is a primary oocyte. So this has 46 chromosomes. Now what am I looking at here? What is the actual oocyte? It's all this right here. Okay? Center white stuff right here. That's the actual primary oocyte. This ring around it, this is the primary follicle. Okay? So this is the follicle around it, called the primary follicle. This is the primary oocyte. Now what exactly do follicles do? They do a few things, but one thing that I would just say that we could all recognize is they help the oocyte mature. Okay? They help it mature along its way. They give it nutrients and things like that. So once that primary oocyte matures, it becomes a secondary oocyte. So this is the same cell we just saw a minute ago, but now a little later in development. It's called a secondary oocyte. This is after meiosis 1. Okay. Now this big follicle around it right here, this is called the mature or the Graafian follicle. This is something you learned back in general NP. Okay. It's real obvious. And what we want to look at here is we're going to take a closer look at these two structures right here that you need to know. The zona pellucida and the corona radiata. These two things you have to know on a microscope and what they are. So these two things, the corona radiata and the zona pellucida, are found around the secondary oocyte, not a primary, only a secondary. And here's a closer look at it right here. Okay. So right here, this is your secondary oocyte, and you have this, in this slide here, this pink ring around it. That's the zona pellucida. What exactly is this? It's a glycoprotein mixture. It's like a membrane around the egg, or the oocyte. Okay. Again, it's a glycoprotein mixture. They consider it to be a membrane around the secondary oocyte. And the corona radiata, these are granulosa cells right here, and they help mature or give the egg nutrients and things like that. Granulosa cells is the corona radiata. So it's kind of a layered thing here. Now, if we go back to one slide, I skipped over this real quickly here. So if we look at this, this is our secondary oocyte. They're calling it an ovum here. But there's your Graafian follicle. So when this is ovulated out, this Graafian follicle here goes on to become this corpus luteum. And I have another picture of the corpus luteum right here. This whole thing is the corpus luteum that I've outlined in white. So this, what is the corpus luteum? Write this down. This is what's left of the mature follicle. This is what's left of the mature follicle. And it's very important. Here's why. It produces progesterone. Okay. If the female is not pregnant, so just let me back up for a minute. Progesterone. When you think about progesterone, what is the importance of progesterone in the female body? What does it help the female with? Pregnancy. Think pro-life when you think of progesterone. Progesterone is all about maintaining this life, this fetus or the embryo. Okay? So the corpus luteum will produce progesterone. If the female is not pregnant, it only produces progesterone for about 10 days. Only 10 days. Because if it realizes that the female is not pregnant, they don't need all that progesterone. Make sense? Okay. If the female is pregnant, though, the corpus luteum will stick around for 3 months and produce progesterone. 3 whole months. Now, hopefully we all know that we're talking about a 40-week birth process, right, with the baby. So what happens at that 3-month mark that the body says, you know what, we don't need this corpus luteum anymore producing progesterone, something else is going to take over for it. What is that something else? It is the placenta. Very good. It's the placenta takes over. At that 3-month period, the placenta is nice and well nourished, it's vibrant, it's ready to take over those responsibilities and continue this birthing or this reproductive process. Okay. Finally, it says, we're asking you here, how many chromosomes would you find for each Without looking at your lab packet, don't look at it, don't look at my PowerPoint. Okay. How many for spermatogonium, oogonium cells, the parent cells, how many chromosomes? 46. Primary spermatocyte, primary oocyte? 46. Secondary oocyte, or the first polar body? 23. Spermatid, spermatozoan, ovum, second polar body? Yeah. And the zygote? 46. Okay, good. Okay, before I hand out the worksheet, what can I go back to at the end of it? What I have here, and I do expect that you get up and look at, these are the things we're testing you on. This is our meiosis board here, if you will, showing you the different meiosis processes here. We've got the egg, or the female over on the left, the male over here on the right. So there's a difference between that. As you can see here, we kind of wrote some stuff down. Only if fertilization takes place, things like that. So I'm going to leave these out as well. Over here we have the microscope. We have the ovary slide. It has a list of the things you need to know here, so you can look at that. Same with this one. This is the testes. And then the last one back here is mitosis slide with the different phases, okay? I'll hand this work